So today is Friday. So Friday is relaxed class. So discussion, no new subject. I mean, I can discuss some extensions of what we have been discussing, but the next subject would be two-species competition, but this will be on Monday. Okay. So I asked you for questions, but you are not very much doing questions and so on. So I have two, but are pretty aren't complicated. So the first question is, I don't know if this is will be covered further ahead. The answer is no. On Tuesday we are talking about modeling biomass instead of individuals. Okay, and we could model many species biomass, say, together at the same time, pretending that they are only one species. When is this case? When is this generalization okay? Okay. So, anyone? So this is from Thales. Any answer? Let people answer. Okay. Yeah, the phone and so on, all this stuff. Okay. Is it?
Maybe it's okay when we are talking about primary production, like microalgae and other primary species.
Could be. More answers. When is it okay? I write down a model. I have 10 plant species, or I have several phytoplankton, zooplankton species, and so on, and I just care about the total mass.
When you are not interested in the interactions between individuals?
Yeah, exactly. Also, but not only the interaction, because you could be, in the case of plants, as I think Mercedes has talked, that sometimes we pretend they are not interacting, because interaction have only effects on a very longer time scale than usually in our observations, and then you use statistical models called neutral theory. But in which case you are trying to look for the species also. But, well, in a sense, I would say, you write models. Models are not like theories in physics, okay, which try to grasp some very fundamental aspect of nature. Models are not like Newton laws or gauge theories or whatever in physics. Okay, so models. When you write a model, usually you have a question in mind. Okay, so if you could be interested in, for instance, fisheries. You could be interested only in how much fish of any species you can get by fishing. In this case, you'd say, okay, I can put everything together. Okay? I don't say that the different interactions and different species are not important. They may be important to determine the growth rate or oscillations and so on. But it could be that you are, at the end, only modeling the biomass. But usually also with things like with plants and so on, when, for instance, you want to know biomass, when you are interested in land cover on semi-arid regions, and you want to know the vegetation, biomass in a certain region, you want to know, am I going to a desertification, or am I stable or not? In this case, you don't care which species. You just don't care. So this is exactly, the answer is, if you care about the species, you have to have the species. If you don't care, because your answer, the species would be not in your data, for instance, biomass. in semi-arid regions, you try to just to say how much you have. You are not saying this and this species, so your data doesn't have the species as only the biomass, then you have only to model the biomass. Okay? So that's more or less the idea. The idea is that models are adapted to your questions and to your data. Okay? Sometimes you can try to write models, but they would never be applicable because they require data that is impossible to obtain, for instance. Okay? This would be a typical situation which is not so nice when you do modeling. Okay? So the answer is this. If depends on your question and on your data. So second one is it refers to where are the refers to those plots I have been doing in the, I think, the first class, yeah? From N and DNTT. Okay? And remember, we could have weird things. Okay? And then we know that if this is the derivative, and I'm here, the derivative is positive, therefore you grow. You increase in N. Here the derivative is negative, you go this way. Here you go this way, here you go this way, and so on. And that was therefore this is unstable, this is stable, this is unstable, this is stable, and so on. You go on exchanging. Okay? So the answer is when f of n, that's the function. Okay? And when f of n goes from positive to negative values, positive to negative values, the fixed point is stable. But when it goes from negative to positive, the fixed point is unstable. Yeah, it's true. Okay? Maybe we can use the derivative of dn dt to find the stability. Okay, the derivative of the function, which would be a second derivative of n. Anyhow, you don't have n, you have only dn dt, so taking the derivative of this. So, and looking if it's positive or negative and determine the stability of the plant. Sure, you can do this. And actually, I didn't do it because it is kind of obvious here, okay? As you noted by the... but just take care. It's not that when the derivative of the function is positive or negative, doesn't mean that there is a fixed point. Here, you see, the thing goes up and down. Therefore, it goes from positive to sometimes goes from positive to negative. So, I mean, this here has more. I mean, you should look at this guy at the fixed point. Okay, you go calculate the derivative, and you calculate it at the fixed point, and then you look if it's positive or negative, and you get the stability.
I think I get the idea, but I think that it should be like dn dt dn, because the derivative is from the... you got it?
Yeah, you are right. You're right, yeah, sure.
Okay.
Yeah. Sorry. Okay, it's dn dt. You should have n. Okay. Sure. Right. Okay. Sorry, confusion. So the point is interesting here is that with single species cases, there's no great point of doing this, because it's more or less obvious that you have a positive, negative, and so on, and blah, 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 blah. But when we go to more species, okay, this will be interesting, because with more species, you don't have this plot anymore. You will have more equations and which are coupled, and they don't have this part. Okay, and then you could go and say, but I can calculate fixed points with two species also. We will do this, and then I want to know the stability, and then you use this kind of thing. Okay, then you use this kind of thing, which will give you at the end a matrix called Jacobian, which is a fabulous thing, but a little bit. You need a little bit more of linear algebra than we are supposing here, but I will discuss it briefly next week. Okay. So that's about the questions, and I was asked if we could have some more examples, and in this case we are talking about examples in the context of single species. Okay, we already discussed the situations where you expect to have single species, and you could therefore let us try to imagine when single species dynamics and the discussions that I've done this week could be, say, applicable. It could be useful or something. Okay, so let us try to remember the main cases where we expected a single species dynamics to be important. Okay, can you remember some of the cases? Yes. Okay. Huh?
The production of beer using Saccharomyces cerevisiae as a single model species.
Yes, this was one. So, and there's another one here. You raised your hand, or yeah.
The generalistic species, right?
Exactly, generalistic species that are preyed upon by generalist predators.
And I was about to ask that, like, how complex the chain that the species are, how complex needs to be this chain for the species be considered and analyzed by a single species dynamics? Like, how many predators and how many types of different resources?
It's not. There's no clear answer to this because all of these are approximations. So it will depend on not on how many predators are there, but will depend on the different attack rates of each predator. Maybe there are many predators, but there is one which is really the one that is important. And in this case, you don't have any single species anymore. But you have a lot, but there's one that's the important guy. So there may have to be many which are really affecting the species. Otherwise, if there's only one, then it's not anymore.
Okay.
So well, therefore, let us put here single species cases that we could discuss. So the first, I would say, laboratory experiments, okay? This would be the cases of E. coli, all these nice guys in the world. The second we have is generalists. I would put in this way from both sides. Okay. Any other case? We have discussed it one more that I would like to.
Invaders.
Invaders. Exactly. So invasion, at least at the early stages of an invasion. Okay, so now we would like to think, so these are the cases where you expect single species to be important, but now from those, which one you think would be better suited to demonstrate logistic growth? Invasion, lab also, yes, when you start with the E. coli, you cover the earth and so on. Okay? So, but here not so much, because there are many interactions. And you expect that this dynamics will lead you, when you go, when you go to the field, you are not anymore manipulating things, you are just counting individuals in the field. Okay? Whatever place of the world you are interested. So you expect this to be more like equilibrium species. Okay, so you expect equilibrium species to be in this case, and then you don't expect so much that you will go on the field and see logistics. Okay, in this case. So you are much more likely to see invasions at the early stage. You see it growing and then stabilizing. Okay, so in this one sometimes will be interesting to look at it as a single species. But this will be related more to theoretical developments. For instance, with network theory, there are many interesting things about this. But you don't expect this kind of generalist, super generalist to appear and grow and then stop and so on. It's much more likely that it is in some, if not stable, slowly varying equilibrium. So, okay, that's more or less what you would expect. And then there's a further point that has been discussed since the beginning of the 20th century. When was the logistic equation written down for trying to describe species? In the first time, okay? So this was Pierre-François Verhulst. In papers written between this and this time. So in the middle of the 19th century, okay? And very nice, but nobody cared, and it was just forgotten. Okay, nobody cared about that. Well, why? Well, not even the concept of ecology was clear at that time. Okay, this is ecology is reasonably. Natural history is very old science, but ecology, in the sense of interaction, species, and the way distinguishing ecology from botanics or zoology or something. Okay, it's relatively new. From the end of 19th century, beginning of 20th century, when it becomes clear the idea of species, the idea of interactions, and so on. So at that time, obviously there were people that were interested in this. Usually it came from demographics, because, for instance, Malthus didn't care about any other species than the humans. He was interested in humans. Okay. Actually, he was, maybe you have read about Malthus. He was not very nice guy. You would not have, if you were a population biologist, you could ask, for instance, if you are a physicist, whom you want to take a photo with? With Einstein, or Newton. If you are a mathematician, Poincaré. And if you are evolutionary biology, Darwin. And if you are a population biologist, okay, you don't want malthus. Why? Well, oops. This is not okay here. There's a conflict between glasses and mic. Competition for. Because, well, Malthus, I mean, he was interested only on demographic, in human demographics. Okay, that's what, and actually then the conclusions come from Malthus, if a population that goes unchecked, that's the name and the use of this word, will grow forever and so on, and it's a problem. So we have to stop it because there will be no food. Okay, and then he came out with all these things like maybe we should avoid poor people having children and this kind of thing, and which goes to eugenics and very, very complicated things. So, I mean, he has made a work that's it's important, but well, okay. Beyond his equation, you don't take photos with him and so on. They choose someone else, okay? So, Verhulst was ignored, but this has been rediscovered by a guy called Raymond Pearl. In about the 20s, okay? So has been forgotten for roughly 70 years, okay? Raymond Pearl was a very important guy. It's one of the fathers of population ecology, population dynamics. He's a lab guy, okay? So, and actually, he was very important also. Well, it's just for some stories. He was very important for the life of Lotka, you know, Lotka Volterra. So Raymond Pearl was a professor at Johns Hopkins in the U.S. And Lotka, you know, the life of Lotka is something very weird. He's an American, but he was born in Ukraine, okay? And was around and studied at UK and initially at Heriot Watt University. So he was more into physics and chemistry actually than anything related to biology. And he was a student of a guy that some physicists will know. Otherwise, this guy is unknown completely, which is pointing. You know, there's a pointing vector in physics, okay? So, and then he moved to Germany. Oh, I forgot the name of there was one guy there that was in chemical physics who was the kind of last person that didn't accept the atomic nature of matter. But he was a very good chemist, and so he has made a lot of works on reactions and, you know, this kind of equations, mass actions and so on, mass terms that comes from him. And so Lotka was very impressed by this. And he said, why can't you, I would like to do this for biologists. Instead of chemical species, I want to do it for biological species. And he went on, and he wrote a book. It's called Physical Biology. Okay? So, but then he went get to US, but he couldn't get a job. Yeah? But he wrote this book, and it impressed very much Raymond Pearl, who called him to work as a non-permanent professor at Johns Hopkins. Okay, so that's the small story. So Raymond Pearl, okay, just for the end of the story of Lotka, you don't have to have pity from him. He became a statistician, and he got very rich doing finance in the early days of statistics. Okay, so he had a good life. So Raymond Pearl made experiments, but it was at the time already clear, and there was some criticism that the experience. I will show you some plots obtained by Raymond Pearl. But there was already a clear a criticism that the logistic is something like this, yes? Logistic curve is something that actually, for many experiments, anything which is like S-shaped like this, okay, could explain the same data, and not necessarily you would have really the logistic, the solution of the logistic equation. Could be something else, because, well, data are usually noisy and so on, and except if you have very good data, you could try to calculate, to see if this is a logistic. But in nature, out of the lab, you usually don't have this, okay? So very few cases you can ascertain that this is a logistic, okay? It is S-shaped and has logistic-like growth, okay? But it doesn't have the status that I want to verify the validity of the logistic equation. So this is something else, okay? So Pearl made, okay, now before Pearl, okay, we have some plots, and I will show you some, which came. The first figure I want to show you, if I can, hopefully work, is, as always, Ecology, okay? So I just took a photo from a book, okay? Which is, if you may want to look at this book at the afternoon or some, which is An Introduction to Population Ecology. It's a classics that's difficult to find around, okay? If the only used one. And this has been written by G. Evelyn Hutchinson, and it's a book that has goes very slowly with a lot of plots, historical things, before the computer was used, okay? So, and also it's pretty much discussing step by step all the concepts in the case of single species and interactions and so on. And so it's very nice. So I took these photos. I mean, I had no other way to transfer this to this, because I don't have a online edition. So this is Ecology, but okay, this was done by McKendrick and Pi. Okay, in this we are talking about 1911. Okay, so nice. Yes, this looks like a logistic, but again, well, could be an S-shaped thing. But you see, with laboratory experience, you have better chances to see a logistic, really. Okay. So then the next figure I want to show you, it will be a figure that I will use next week again, is Paramecium. Paramecium is a protozoa that you can grow in the lab, and that's very important in the history of competition theory and so on. We will discuss this Monday. So this has been experienced by Gause, who in the 1934, and Gause was from the Soviet Union. I think he was also Ukrainian, and actually Raymond Pearl also invited Gause to Johns Hopkins at the time. So, and so here you see growth curve that looks like a logistic. Okay, so this is what I would call generalized way logistic growth, even if we don't know if it's exactly the logistic. But obviously, if you in order to determine K and R, I mean K and R are easy. Yes, how would you determine K? I mean, it's written there. Yeah, you just go and look if you get to a constant value, and then that's K. Okay, it is. You could argue it's not so constant. There are some of them outside. Maybe if you went more, a week more, maybe it would happen something. Well, we will never know because it's only 7 days, 6 days. Okay, so how would you determine R? I mean, the logistic equation is like this, yes? So this guy from experiment, it's easy. Okay? How do you determine R?
Fitting the curve?
Hmm?
Fitting the curve?
You can fit the curve, yeah, sure. Yeah. So near the starting here, the population is small. Okay? And being small means that this is practically equal to 1. Okay? Because this is small, n over k small. This is practically equal to 1. And so at the very beginning, you have only this thing. Okay? More or less. So n would grow with e to rt. Okay? And then you could, for instance, go for your data, and you would maybe take the logarithm. Okay, so let me write this. Of n is equal to logarithm of n0 plus rt. Okay? You put it on a plot. Logarithm of n in time. And this has to be a line, and the inclination of the line is r. Okay? So you use both. In this case, you need only to use the both asymptotic cases, near 0 and near infinity, which in this case is 6 days. Okay? So this is how you would get the parameters of the logistic, and it's relatively easy. Okay? So next. So next thing is okay, Raymond Pear, which was really interested in knowing how precise the logistic was. I mean, it turned out that all of this discussion until the middle of the 20th century ended up with this. Logistic is interesting. It's a conceptual tool that we can use, but it's usually difficult to state that it is exactly that equation. Okay, but anyhow, it's useful. For instance, we can talk about carrying capacity, which is it's for now. I think everybody has some intuition of what is the carrying capacity, and it has introduced it to the logistic. It was the easiest way to think about it. Okay, so can you see? These are the experiments by Raymond Pear with 3 types of experiments with this Drosophila melanogaster, which is fruit flies. Okay, so you see, depending on, I mean, one is the wild type in a pint bottle, then there's a manipulated thing, and then there's wild type in a half pint, okay, which is and here you see that the kind of things that you obtain. What would be interesting? If you look at C, that is the same species as A, okay? But this half of the volume of the experiment, okay? But the K is 212, and the other is more than 1,000. So the doubling the volume it did not double the carrying capacity. It was much more easier. I don't know how the control of this experiment is, because it depends on how you feed them and so on. So, but there's the thing. Then you come again to the questions that we have already discussed, which is the nature of the carrying capacity. When you now try to see, okay, why is the carrying capacity, why does it have this certain value? Well, that's not so anymore so easy, because then you really need much more biology, much more processes. What makes the population, what affects the carrying capacity more? What makes it grow or not? Okay, so then you cannot do anymore this kind of general theory that we are doing. We are speaking from zooplankton to elephants. Okay, and when you want to go for this kind of thing, the values of the parameters, then you need much more biology. And I don't know exactly the explanation, and I didn't see a discussion for this, but anyhow, it is interesting to note that the carrying capacity is not trivially. I mean, I give you twice the territory, there will be twice carrying capacity. You have to verify it for the species you are working in, which territory you are working. Okay. So next figure. Yes.
You repeat, like, the difference between the lines are not in the species per se, is in the environment?
No, just
no.
The B is something engineered, not genetically, but it's something, forget about B, okay?
B, okay.
Other guys. C and A are the same species, are wild type Drosophila melanogaster, okay?
Okay.
And one is, this is done in a kind of bottle, okay? And you have a certain volume for C, and you have the double for A. Okay, just the experiment has the double of volume. Okay.
Okay.
So, and you see that doubling the volume does not imply that you have the double of the carrying capacity. It has much more. Okay.
Okay. Thank you.
So, let's continue our show of photos taken from a book. So, next interesting thing is okay. Next is just a very famous experiment. Where's the dove? The color dove. Okay, this is it's a dove like this one. Where's the dove? Okay, it has a collar here. Okay, and it was actually a continental species in Europe. I think more to the eastern Europe, but that at a certain point it started to invade, and then invaded the British Great Britain, and it was observed by many. I mean, there are bird watchers and so on, and so they collected a lot of data. And you see, it started in 55, and the data collection went from 55 to 1970. You see a nice growing curve, but will it be as logistic or whatever? You never know. But this is an example of invasion. It's not lab. Okay, that's why I'm showing you. That's an invasion. And in invasion, you have this growth, and somehow it will stabilize. Okay, that is the interest of this case. Okay, that's a real ecological thing. It's not a lab. Okay? So another case, I don't have the figures here, and I don't know how to find them. Is also invasion of a kind of rat, but it's not a rat. It's well, okay. It is a big rodent from the Americas, which was hunted for. I mean, not for using parts of the body to produce perfumes and this kind of muskrat, okay, and that was taken, and then, as it always goes, somebody said, okay, I will take some to my place. In this case, Prague, in the Czech Republic, and we go in there. I don't have to go hunt them over the Atlantic, and yeah, sure. But they obviously escaped, and they found, oh, and we like Europe, and then they invaded all of Europe. Now they are widespread, and then you have also this growth and saturation. Okay? So, next figure will be more interesting. So this is all these are the nice stories of logistic equations and so on. Very beautiful. But let us see something more. So there's another Drosophila kind, which is this one, which is called Drosophila virki. So you have they have two different populations. And this is a work by Ayala in the 68. And what you see, this is pretty much more noisy than we had before. It's a long time experiment. It's weeks. Okay? It goes to 70 weeks. It's more than a year. Okay? So long experiment. That's because it's that's why it's important. Okay, because growing flies like pearl and so on. Well, okay, nice, and everybody at a certain point knew how to do this. Why would a person back in if pearl was 1932, more than 30 years ago? Again, but it's a little bit different species, but. Again, grow and see the logistics and so on. But so the reason is it's a long time experiment, long time for Drosophila case, okay? And this is really something that you could take at one of the take-home messages from the course, from the more practical point of view, is it's really precious if you have long time data in ecology. It's rare, it's difficult because. You know, even if you plan a long time thing, and sometimes, I don't know, after 2 3 years you have fire, you'll lose your grant, you cannot continue or something. So it's not an easy thing to have long time experiments. We have a very nice long time experiment in the Amazon, which is the project of biological dynamic of fragments. Near Manaus, which is an initiative of the National Institute of Amazonian Research, which is INPA, and the Smithsonian. And this is going on since the 70s, I think, something like that. This is a very nice thing. You can go and study plants. You can go and study long time census of certain species and so on. It's really something very, very nice which we have in Amazonas. So okay, so back to this. It's a long time for Drosophila, and what you see here is okay. They start growing, and then you see they oscillate quite a lot. Yes, but okay, you take more or less you try to fit a line through the part where after the initial growth, you try to fit just a straight line. Okay, so fitting is about this. You try to fit the best line, and you get these ones. Okay, so what's curious about these lines is that they are not constant. They are slowly growing lines, which would be translatable as maybe the carrying capacity is augmenting, is increasing in time. Okay, on a time scale, which is not the time scale of the reproduction of Drosophila. It's the time scale which is very long for Drosophila. It's more than a year. There are many generations here. Okay, this is not 1 or 2 generations and so on, because they don't live, I don't know how long they live in the lab, but anyhow, it's not even a week most of the time. So you go on generation by generation, and so you have this long time thing. And it seems that somehow the carrying capacity is growing on the long time scales, okay? Many things in modeling and in ecology, we have to think about the time scales. Okay, you have a long time scale and a short time scale. If you want to, what exactly is the time scale? So it's a scale. It's not something that you have a value. But long time in this case is many weeks, and short time is 1 or 2 days. Okay, so on the long time it's growing. Now, very nice. Explain me why. Evolution.
Maybe I think there is some kind of evolution.
Yeah.
Maybe the individuals are getting smaller, or somehow they are evolving in a way that they need less resource to survive.
Yes. Okay, you found it immediately. It's more technically, I mean, there are sometimes it's a kind of it's selection, okay? Of what? There's a large genetic diversity, even being the single species. All of them are the Drosophila birki, but there's genetic variability, and the genetic variability will not be important on the short time scale. Okay? You don't have time to accumulate favorable mutations that would increase the fitness of some individuals that have this mutation. But over the longer time scale, you have diversity, and then you get actually some kind of competition between different subspecies, let's say. Okay, it's still the same species, but there are mutations, and then you have the competition of the mutations that are expressed as phenotypically as parts of the individual, which will create this kind of intra-specific competition, but between different, say, strains, if you want, of the same species. So you get the competition, and you have a question.
So in summary, in the beginning, in a short time evaluation, it's approximate to a logistic model because the interactions, the competition is too slow.
Yeah.
But as the resource and the curve arrive the carry capacity, these interactions and competition is too high, and the variation became more increased.
Yes. So that's a very good point. That's what you just said what I was going to say. So at the very beginning, populations are small, and genetic diversity, variation of who is the best competitor. Okay, it's not very important because competition depends on the amount of resources. Okay, if you are in a situation where you have plenty of resources, then what happens? You just grow. Okay, and at the beginning, you have also the genetic diversity. Okay, so at the beginning, the subspecies, the strains that have larger R, will dominate because they grow faster. Okay, and then they come to a high value quickly, and if the genetic the subspecies that have a smaller R will lose the race. It's not the competition, but it's a kind of race. Okay, so the race is such that if R is those species or subspecies that have a larger R grow and dominate the situation, and you will see more of them. But then when you come to a situation where you are pretty much at the carrying capacity, nobody is actually growing a lot, okay? But that means, I mean, they're still going on birth and death, but okay, but it's at a sustainable level, and there's no big point of being a subspecies with larger R. But it's a very good point to be a subspecies with larger K. So larger K is you are better in competing. Therefore, you can adapt to the situation. Depends exactly on what. Maybe you are better at food consumption, resource consumption, or whatever. Then you prevail over the others, and then you see this kind of thing. So there was a question here.
Is this the reason why the carrying capacity did not have relation with resources quantities in
the previous
graph?
kind?
Yes.
I don't think so, because that was not a long time experiment.
Okay.
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 So in order to see this, you have to. This happens on different scales. So now you could think about the same situations, that not with a single species, but many species. Okay. So this is, for instance, if you have.
Sorry. So in this example, we don't have a constant R, or
Well, R from the logistic is a constant. It's the initial growth rate. Okay.
yeah.
The initial growth rate, it's a constant R. The growth rate, the effective growth rate at a certain point, well, here it's 0.
Yeah.
I mean, d and dt is the growth rate. Okay. R is called sometimes the intrinsic growth rate. So that's the growth rate at a very small population. R is a constant.
Okay.
Okay. The growth rate is just like this curve. This is d and dt.
Yes.
Okay. That's the growth rate. Okay. It starts. No. What are we? No, this is n. Okay, sorry,
Yes.
this is n. Okay, so d and dt starts increasing exponentially, then it slows down and goes to 0, d and dt, okay, at the end. Okay, so.
But when we start to see the increase in a line, we don't have a logistic model. So I mean that we have to change the model, or.
So you can do several things. You can do it by saying, okay, I observed this. Then you could say, I could write down this equation, okay? And put down a variable carrying capacity, okay? But that is much more complicated equation than the previous. And then depends on how K is dependent on this. It's a given function. Okay. Then you could have this. This could give you very complicated dynamics. But in order to mimic what we see there, you need that the K is variable on a time scale. So the growth time, remember, R is 1 over time. So there's an intrinsic, so 1 over R has dimensions of time, okay? So this is the intrinsic growth rate at the beginning, okay? Then K should be evolving much slower in a time scale, okay, that is much longer than the time scale of the. Then this equation will have as an approximate solution something of that kind.
Okay. Thank you.
Okay, Michael. That's it, somebody else.
K could be dependent on R because
What?
on R.
Could. I mean, could be trade-offs between R and K. Trade-offs. It would be an evolutionary thing, okay? But could be, but in evolutionary sense, I mean, long time sense. Yeah. So, okay. So, in order to check this speculation, actually, Ayala.
Being K fixed and stable and attractive fixed point, why don't we see all this fluctuation? Like, why would the population decrease after reach K?
No, why would the population decrease?
Yeah.
After?
After reach K, like.
You mean this up and down here? This is noise.
Yeah.
The environment is never constant.
Okay.
Yeah? And environment is never constant. And actually, we are making an approximation that population homogeneity, actually. That's that. Okay, so we now discovered that it comes as an effect of heterogeneities, genetic diversity, that maybe K is variable. But then you see, we are not fitting exactly in a curve. It's a very rough approximation, okay? So you go up and down and so on, but over a long time, there is also variability in birth rates, death rates, and the environment to keep. I don't know back in the 60s how constant is the environment. Okay, so you have birth and death rates fluctuating, and then there's a lot of things going on. Okay, so it's not that easy. I think I have even another experiment, if I'm not, where the people try to find something like this. Let me see. So this was an experiment, and you see, it's 10 years experiment of woodpeckers. I have even a photo of a woodpecker here. Yeah, sure. Very nice. Yes. So there's a specific kind of woodpecker, which is it's in New Mexico, in United States, and you see that's the population census. So you don't have, in this case, this bird has been there for already a long time. So there's no initial growth. It's not invasion. Therefore, you expect to be more on the carrying capacity at the time. Okay, but you see, there's a lot of fluctuations going on. And actually, if you try to draw the best line above, it's a little bit decreasing population. Okay, but this is in the field. Now, obviously, in the field, there's much more variation than in the lab. Okay? So in the field, there is no such thing as a really single species population, as you could have in the lab. Okay? So there are lots of interactions and so on, and you try to more or less think it as a single species, and so on, and you have a certain level of what should be more or less the carrying capacity of this bird in that region. But you see, they also looked at the survival from year to year, because they breed once a year. Okay, and they have looked the survival of adults and juveniles. It oscillates a lot. Okay, and also the number of offspring per female, or per pair, if you want, because they have a 50-50 sex ratio, is also oscillating a lot. And this is due to the fact that you have many kinds of noise. Okay, so do you have what kind of noise would you have? What kind of stochastic effects would you have in this case? Now, one thing, this is in nature. It rains, it's climate. There's human interventions people building houses or doing whatever, I know. So there are lots of things that are non-constant and more or less, you can say these are things that happen randomly, therefore there's a randomness there. But then there's a second kind of so this would be environmental noise. You will see this with the next course with Ricardo. But this is environmental noise. You could also have intrinsic noise of the dynamics, okay? So coming from genetic variabilities and so on, things that are not taken into consideration in a single species or even a multi-species models that do not address the genetic composition and do not address fitness of different individuals and so on. So there's a lot of things going on. So you see, it's actually there's a lot of variation here, but actually population is more or less, it's not constant, but does not oscillate as much as the survival and the number of offspring per female. So it's also interesting. It's not even with a lot of diversity in both of the survival and fecundity, the population is, but it's decreasing in this case. Okay, so then, just to go back to again to the.
Sorry.
Yeah.
I have just one question about the modeling. Like, the K would variate. Like, how could we improve the model? How could we discover the constant in which K is increasing and improve the model?
So okay, this is the thing that you would do. It's the simplest thing. You just have to have that the time scale of variation of K is slow, and that's it. And you mimic your results. But then again, you could say, okay, but we want more. We want to have, as I told you, K is a phenomenological constant, or even a phenomenological function in this case. We could want more. So we could have population dynamics where you would have to do this in a usually you do this with stochastic models, which will be discussed by Ricardo, but where you have individuals and they have traits, okay, which are expressions of their genome and variability and so on. Then you can model selection of traits in populations. And that's the way that the things are done. It's a much more complicated thing. It's actually things that are being published, because this is, again, it's long time, blah, blah, blah. So a colleague of mine, my former student of mine, had actually worked on that, but with people from Los Angeles, UCLA, where they have a lab with fishes, and several species and so on, and this goes on for years and years. So they have data, really rich data, and they also know better which traits are being selected, because it's difficult to know the traits that are being selected by Drosophila melanogaster.
It's working now. Okay.
Yeah.
Just checking if I'm correct. If we could discover how K it's increasing, we would know how it's going on between the selection and traits, and how the traits would evolve in the population.
Yeah. So this would be a mixed population and population genetics, population in a single species kind, or maybe interacting kind, which has a new component, which is the genetic variability. So you have population genetics also. You have to put this in a model, and then try to see if this can explain experiments. It actually can. But I mean, I know of this work published like less than 10 years ago, so like 6, 7 years ago on this kind of thing. But these are complicated models. Yeah. Yeah. Okay. No question.
I have a question because I remember the last class that you told about co-webbing. This phenomenon could be like this oscillation around K and not a noise?
Oh. Well, you don't have any reason to think that this is only due to density dependence. Okay? So remember, we had this oscillation, cycles of 2, okay, for instance. So it was oscillating. So you could say, maybe this is not a constant, actually this should be an oscillating thing. Well, again, then you have to think about, is it biological reasonable to think about this, okay? Because what would be the mechanism that would induce the oscillation? And this is always, when you go in nature and see oscillations, first you have to be sure that it's really oscillation, it's not noise. And then if you're sure that there's a really oscillation and not noise, you want to know the mechanism, and there are several mechanisms, okay? And in this case, I cannot think of any really important mechanism that would make this oscillation happen, as these are not non-overlapping generations, and actually what's going on is not blah blah blah. It's not, and so I don't think it would be, because it's not that you could not mimic this behavior. You could mimic this behavior, but there's no biological clear explanation to use, for instance, a logistic discrete time in this case. Okay? And moreover, it's not only it's oscillating sometimes, but the trend is decreasing, and then we didn't see this in the there, yeah? Or increasing. This in one case increasing. Yeah.
Hello. Okay. Also, I'm just trying to make sense of the relation between R and K, because in that experiment, for example, I don't know if this paramassing one, you like increase the media, and the K goes up, and the R is the same.
This was not the parameter. That was the Drosophila.
Oh. Okay.
Yeah.
So
Melanogaster, not this last one.
And then, so you can like increase the resources, the K would go up. But there is the R would not change because it has to do with the metabolism and so on.
Yeah.
In the continuous model, R and K, they are like separate things. In the model, you can change one without changing the other.
Yeah.
And the one with the delayed time, eventually, what happens is that if you change R, you will change K.
Yeah, I mean, this. Okay, let's talk about something that I make it clear. There's something which is one of the very first discussions about evolutionary trade-offs and so on, which is called, was a debate. This is the RK selection. And this is related to situations like, we are talking now about multi-species, okay? But it could be strains of the same, but okay, let's think. So sometimes you have the situations like that your habitat is initially, say, destroyed. Fire in the forest, for instance, okay? And then, well, then it will go back. I mean, it's not completely destroyed, let's say. Okay, it's very strongly perturbed, and the most species that were there aren't anymore in a certain restricted area. Then you have new species arriving, okay? So this new species will be the situation of an invasion, and the species that will be successful in this invasion are species that have the larger or the bigger R, okay? R is big, okay? So there will be several of them competition is not very strong, but there is a kind of competition also because, I mean, you will have much more over some generation, much more of one of them than the other ones, okay? So there may be the residual individuals of the others. It's not complete elimination, but then you have, so the metabolism has to be such that they produce a lot of offspring. And usually species that produce a lot of offspring don't have parental care or something like that. Okay, there's no, that's a technical term, parental care, more for animal ecology, but okay, could also be applied to plants. So, so most of the offspring is not viable. Okay, but there are so many that at the end you have a lot of offspring that are viable. Okay, that happens a little bit. Fish also, that put a lot of eggs and only some of them go. It is very common also with plants. You have lots of seeds, but only some of seeds will grow. So this is kind of what people say. These are called R-selected species. Okay, R-selected. Now, this will grow, and then we will have real competition for resources. Not only, so when you have real competition for resources, those that are there and there will be this kind of selection to the best competitor. And the best competitor is the one that has better competitive skills, and some depends on the exact nature of the competition, but that will increase K. So we'll have a larger K for this species, and then this species will eliminate the other ones. So when you have this kind of situation, these species are called key selected species. Now, these species have no reason to have a very large R. They don't need a very large R. So there's this kind of relation. It's from selection trade-off. Okay, the R selected species, there's no great point of being very competitive. You will not invest effort or energy in being very good competitor, because then you probably have a lower R and then you are eliminated. So put all the effort in growing as fast as possible, R selection. Chi selection would be put all your effort on competing, and you don't care about R. It could even be lower, and then usually these are larger species. For instance, larger trees. If you have like in tropical forest, you have some fire, and then first you have smaller trees. It's not that immediately you get back to the nice big trees. They are K-selected. So first you have the growth of some species, and then you have the invasion of the key-selected species. So that's called ecological succession. Okay? And so the relation ends up being an evolutionary trade-off of species that have very good competitors. Usually they have, if you think about animals, they have parental care, like humans, but many big mammals have parental care. Okay? And maternal care also. I mean, there are many things like that which will be connected to less offspring, but taking care of the offspring and make every offspring viable. This is usually connected to a better competitive ability, and this will make the key selection. Okay? This is a little bit outdated. I'm not an evolutionary biologist, but I think Flavia, probably it's not around, but she knows everything about this, because this has been, I mean, it's not anymore the thing that people do nowadays, that back to the 50 years ago was. But anyhow, it's a very simple explanation, but it's nice to see how with very few elements, just R and K, we can discuss succession in several situations and so on, and connect this to real species and try to understand what's going on. But this is a little bit also the reason for models and for theory. Okay, so theory. Sometimes you could like, I don't want theory. I'm field ecologist, and or I'm field epidemiologist or whatever. Don't need theory. Go just collect the numbers, okay, and look tendencies. But you will be. What would this mean? Okay, you would like to trends and so on, but you need explanations for that. Okay, therefore you need the theory. Otherwise, you are collecting stamps. Okay, it's nice to collect stamps. You don't need any theory. Just beautiful. Okay, but okay, you want to do science, you need some theory. Even it's not a mathematical theory, it could be a conceptual theory, but you need a conceptual framework in order to discuss things and even to propose experiments, because you cannot go just collecting all data. You have to know what data are important, because you have to have an experimental design, an observational experiment in the field or whatever. For instance, in public health with epidemiology, you have a very complex system with humans that interact, and then you have the way the data is collected. All of this is very complicated and so on. But you have to discuss your problem, maybe the population levels or oscillations or whatever, from a viewpoint that has at least behind, if it's not a model, it's behind, there's a theory for that. Okay, and that's the real point about this kind of things. It helps you to formulate problems and make hypotheses that you can eventually test. Okay, not so much. So that's why I'm not talking so much about I want to fit exactly this model to some data. Okay. The status of modeling and theory in ecology is not like in physics or chemistry. So just to finish, there's a still nice, picture, which is continuing the discussion about this Drosophila Birki by Ayala. So he went on to test the hypothesis that genetic variability is in somehow related to the increasing carrying capacity. So what he did is this here. There's a new experiment where there were 3 experiments. 2 of them, the one below is just the same kind of thing he had before. You see the tendency a little bit increasing. So the other species have been irradiated. Okay, so irradiates the species. Okay, what happens at the beginning? They suffer. Irradiations makes R smaller. Okay, but well, after some weeks, this irradiation is already diluted. There's no point anymore. So they will grow, and actually they grow much more than this one. Well, what has happened here? But irradiation created a lot of genetic diversity artificially. Okay, and then he's shown that the genetic diversity gives you a higher K. That's a very beautiful experiment. Yeah, I think everybody will like this experiment. This guy is smart. Yeah. That was really good, okay? Yeah, he should have a prize for that. And this was back also in 1968. Yeah? Very, very interesting. If not a demonstration, a very plausibility argument for it. Yeah.
That like if we are considering like fierce selection in the end, like with the car, we are considering that we will have like a less genetic.
It's oscillating chaotically.
Okay, it's back. So we are considering that we are having less genetic variance in the species, right? Because we had a lot of variance, and then...
In this case, you mean?
Yeah, not exactly. Like when we are in the stabilizing region.
Yeah. No, you have still, I mean, you have still genetic diversity. Right? It will always have, okay? And when you're near K, the genetic diversity, which affects K, because you have also in the beginning genetic diversity. But if it affects K, it's no point. K is not important. R is important. Everything that affects R is important. Then everything, when you are in a stable or more or less stable situation, every genetic variation that affects K will be important. So you have still going on, okay? And then it will depend on if this converges to something, if it's a convergent selection or not. And this will depend on the species, on the situation. So could you maintain the pool of genetic diversity, or will you converge to something? Okay, that you can ask to Flavia, because she knows everything about convergence and non-convergent diversity. Okay? But you could have both cases. You can have a convergence or not. Okay? And in convergence, it would mean easier to be endangered, because everybody being very similar. If you have a pathogen, everybody dies from the same thing.
What were the details of this one? The species and the kind of radiation
This
they
is
were exposed
resolved. The kind of radiation, I don't know. It's not in the book, so this is kind of thing that it's difficult to obtain the paper. I just got it from the book. And the book didn't tell us how they irradiated. But the Drosophila, this is the Drosophila birki. Okay. It's the same that we had before.
The same.
Yeah, that's, I think it's in maybe the subsequent paper from the same group, Ayala, and then just to prove the case that genetic variability is involved, they created artificial genetic variability by irradiation. But I don't know how this is.
And those three groups were all the same species?
It's all the same species. All of them are Drosophila birki. Okay? So, we are almost at the end, and I will just talk about a little bit something else without any great models, but I want you to remember of what is an Ailey effect. Okay? Remember that when we discussed the continuous time dynamics, we could have things like this one, dn dt as a function of n, and I have drawn this thing, okay, which stabilized the zero, okay? This is stable, unstable, okay? So, just to put words on this, here, when you look, so one equation that would be like this is just R and our usual equation with an a plus with something here, a. Okay, this is a cubic polynomial, and it would have this. You can plot it and so on. So, just this means that when you don't have this here, all nonlinearity density dependence is such that increasing n decreases the growth rate. Okay, so it's called negative density dependence. But the effect of this one is a positive density dependence, in the sense that you have more, then it grows. And it dominates this part of the system, and this other one dominates that part of the system. Okay? So, just ideas about what could create an Ailey effect?
Interbreeding, because you have low population, they are going to breed between themselves.
It could be
genetic diseases are going to appear more often.
Could be interbreeding. Yeah, another.
A kind of behavior that needs a lot of group protection or something like this.
For instance, we have I even have a plot somewhere from species that when they are larger groups, they get less predated. This happens with fish, birds, and I had even a plot, if I don't know anymore where it is, of the caribous in this is caribous, you know. We have a photo of caribous. It's tundra species, yes. This is a caribou. Okay, so this is these are observations made in very cold regions of the world. I don't know where this was exactly, but so this is the per capita population growth rate. Okay. So you see that it's negative if the density is small. The density is small, you have the same area, density is small, it means less individuals. So it is even negative, and then here it gets positive. So you need this threshold. So this is the example of a threshold due to higher protection. And then it could also have the predation, which would be a collective behavior that enhances predation capacity. That is a kind of situation where, which happens, for instance, it's called cooperative hunting, which hyenas and actually wild dogs. Okay, they have to form groups. If the group's too small, they cannot survive. So what is behind this example, except the one about interbreeding, is that this is the first glance that you can have in models that have not only competition, they have cooperation. Okay, so you are modeling cooperation in this case. Okay, so intra-specific cooperation. Okay, they need to kind of collective behavior. And in cooperation, obviously this is a concept, ecological concept of cooperation. It doesn't need that they don't have to cooperate in the sense that humans would say, oh, let's do something together. Even bacteria could cooperate in this sense. Yeah.
The transmission of a resistant gene through plasmidial portion of some bacteria to another, and resistant against a bacteriophage is also an example.
Yes. There's also with, for instance, fish, goldfish. You know, this little nice goldfishes. So they also need a minimum density because they release chemicals in the water, and it's calcium or something like that, which actually harms the possible predators. They go away. Okay, but if it's too small a group, that gets diluted, and then they are exposed to predation. So they have to do this. So it's releasing chemical elements in the water, which have to have a threshold value. Right? Yeah. And so to end the discussion, I was talking about goldfish, and this was actually, there's an interesting case about a goldfish that has been experimentally studied by Ailey back in the beginning of the 20th century. And there's a point. Now think about fishes, not necessarily goldfish, and that you go and harvest, you go fishing. Okay? So if there's a species that has no Ailey effect, okay, you know that you can overfish. Okay? You fish so much that you induce a death rate, artificial death rate. And eventually this could be higher than the birth rate. Therefore, you will have smaller and smaller populations. And that's it. Okay, that's the whole story. You can overfish. Okay? But if you overfish, then you stop overfishing unless you have really eliminated it. You will grow again, and then you can get it back. Okay? But if you have an Ailey effect, you go on fishing and you see the decline in population. But maybe you arrive at this point, and then you collapse immediately to 0. And then you say, oh, sorry, let's stop. No, you are not tractor. This is a catastrophic transition. If you have, it's not non-reversible transition. We will talk at the end as examples from fishing and from vegetation dynamic desertification. These are called critical transitions, because if you have this Ailey effect and this you are harvesting this population, what you will see is that you induce a death rate, therefore the population gets smaller and smaller, but if you come to the threshold, the population will just die out immediately. It's 0 is the fixed point, and then you lost it. Okay, it's not reversible.
Sorry. So how we can calculate biologically the A, because it's not like capacity de carga.
Well, it's not it's a threshold point. It's not carrying capacity. Well, this is you have to go be species specific. I mean,
okay.
what? So there's a book here that you may like to take a look. Later, it's called Ailey effects. A whole book without almost no equations, just all the kinds of situations that induce Ailey effects, and then the threshold value will be determined by the biological details of the interactions going on. Okay. So, okay, thank you.
So,
more questions.
Yeah. In the model, there is no Ailey effect, and using this example that you've mentioned about a death rate that induce it, like if we induce a big death rate, but it will make the competition get slower. Yeah. So it can be a manner to induce a bigger K because you have a bigger diversity because the competition is smaller. So
you could do selective fishing. Yeah. And induce different kinds of variabilities. That is true. I mean, I'm taking here the case without any genetic variability in an example.
So it will reduce the genetic variability and will increase the K, right? Eventually.
On the long time scale.
Yeah.
Long time scale. And then you have also situations where where you have species that have something which is called ontogenetic shift, which means that they start at an egg, then they are small fishes, and then they are big ones. Yeah. Maybe there's even cannibalism, and there's a whole thing going on. There's a whole book, I can bring it to you next week, on population dynamic with ontogenetic shift, which is very important to explain, for instance, the disappearance of codfish in the Baltic Sea, which is a disaster for them. The codfish is a valuable fish for consumption, disappeared by overfishing, but it was not clear what the mechanisms were, and they are related to this kind of interactions where the mechanisms that protect or species are very different in the different phases of their lifetime. So, thank you. We will continue Monday. And now we will go to two-species model, and we will discuss competition and predation. We will not discuss mutualism, because mutualism is not so interesting with two species. Mutualism is nice when you have networks, and we don't have it. And so that's the next thing. Okay, see you Monday.